In this video, we will study about nucleosides and nucleotides. Nucleosides and nucleotides, they are building blocks of DNA and RNA. Full form of DNA is deoxyribose nucleic acid and full form of RNA is ribose nucleic acid. Now, nucleosides and nucleotides, they are made up of nitrogenous bases. Now, nitrogenous bases are of two types, purines and pyrimidine. Purines are further of two types, adenine and guanine, and pyrimidine are of three types, cytosine, thymine and uracil. Uracil is part of RNA and thymine is part of DNA. Now, if you see the structure of pyrimidine and purine, pyrimidine is made up of hexagonal ring. Now, this is thymine structure, this is the structure of cytosine, and this is the structure of uracil. Purine is a 9-membered ring. Now, this is of two types, adenine, and this is guanine. Now, second constituent is sugar. Sugar is made up of 5 carbon, so this is called as pentose sugar. Now, this is beta D ribose, this is beta D2 prime deoxyribose. Beta D ribose is constituents of RNA. Beta D2 prime deoxyribose forms DNA. Now, if you see the structure, now this is the first carbon, second, third, fourth and fifth. And second position it has H and OH. But if we compare these two sugars, here at second position this will have H and H. Oxygen is not there. So this is called as beta D2 prime deoxy, mean this is without oxygen, beta D2 prime deoxyribose, which forms DNA. Now third is phosphoric acid. Now this is phosphorus, OH group, OH group, OH group, double bond O, and when this bond weakens up, this can have negative and positive charges. So now, what are nucleosides? We can see here, this is the nitrogenous base. It can be any of the nitrogenous base. It can be purine, it can be pyrimidine. Now, this will combine with sugar. Now, sugar can be deoxyribose. This can be ribose sugar. And when these two things combine, now here water loss occurs. This hydrogen and OH group, they combine to form water. So, a bond will be created between the two. Now, this bond is being created between nitrogenous base and sugar. So, this is also called as glycosidic bond. So, this combination of sugar plus nitrogenous base is called as nucleoside. So, what is nucleoside? Nitrogenous base plus pentose sugar. This is called as nucleoside. Now, if adenine combines with ribose sugar, this will be called as adenosine. And if adenine combines with deoxyribose sugar, this is called as deoxyadenosine. If guanine combines with ribose sugar, this is guanosine. And if guanine combines with deoxyribose sugar, this is deoxyguanosine. Uracil combined with ribose sugar, this is uridine. And thymine combined with deoxyribose sugar, this is deoxythymidine. Now, what are nucleotide and what is the difference between nucleosides and nucleotide? In nucleoside, we have only two things, nitrogenous base and sugar. But if phosphoric acid also combines with nitrogenous base plus sugar, then this becomes nucleotide. We can see here, this is the nitrogenous base. It can be any of the nitrogenous base. And this is the sugar ribose. It can be deoxyribose sugar also. This bond is glycosidic bond. And this is phosphoric acid, which is combining at this 5 carbon of the sugar, and this is OH group and H. Now they again will lose a molecule of water and a bond will be created between them. This is called as ester bond. And this sugar plus nitrogenous base, any of the nitrogenous base, whether it is purine or pyrimidine, and this is phosphoric acid. So this forms nucleotide. Now what are nucleotides? If we have deoxyribose sugar and base is adenine, this is phosphoric acid, this is glycosidic bond and this is the ester bond. This is called as deoxyadenilic acid. And if it has guanine, now this is guanine, ribose sugar, deoxyribose sugar, phosphoric acid. So this is deoxyguanilic acid. And if it has cytosine plus deoxyribose sugar plus phosphoric acid, this will be deoxycytidilic acid. Thymine plus deoxyribose sugar and phosphoric acid, this will become deoxythymidilic acid. Uridilic acid is absent in case of DNA. This is having only thymine. Now, if sugar is ribose, there is sugar, nitrogenous base, and phosphoric acid. This is called as adenylate acid. Again, if we have guanine plus sugar plus phosphoric acid, this will be guanilic acid. If we have cytosine plus sugar plus phosphoric acid, ribose sugar, this is cytidilic acid. And if we have uridine plus ribose sugar plus phosphoric acid, this is uridilic acid. So these are constituents of RNA. And when deoxyribose sugar is there, so they are constituents of DNA. So when a nitrogenous base plus sugar plus phosphoric acid combine, this is called as nucleotide. But if we have only nitrogenous base plus sugar, this is nucleoside. So this is the difference between nucleotide and nucleoside, but both are building block of DNA and RNA. Thank you for watching my video. If you like my video, please like, share and subscribe.